We are thrilled to welcome Peter Smith, co-founder and CEO of Remix Therapeutics, to the show today. Thank you once again for joining us.
Yeah, thanks, Drew. Great to be here.
It's amazing to have you, Pete. I'm so excited to dive in here sooner. Really, just to kick things off, Peter, could you just share a brief personal intro with us?
Sure, yeah. So again, great to be on the call. I'm looking forward to telling you more about Remix and what we think about this very exciting space of small molecule and RNA biology. My background, I'm a cancer biologist by training. Started my training at one of the Cancer Research UK sites in the United Kingdom. Got really interested in oncology, drug discovery, and how to take molecules forward and help patients. And so with that as kind of my PhD that was co-funded by Eli Lilly, and seeing what drug discovery really looked like really inspired me to want to build a career that was making new medicines for patients, primarily in the oncology space. So I eventually moved over to the US around 20 years ago, where I did a postdoc at Dana Farber Cancer Institute in lymphoma biology. I had a fantastic time there, but always had a lens towards wanting to move into industry and work in drug discovery to translate those academic learnings that I had into real therapeutics, and was fortunate enough to get a job at Millennium Pharmaceuticals in the oncology discovery group. I was there for about 8 years in the end. It was half Millennium, half Takeda. Takeda came in and bought Millennium, and really had a fantastic time there working with some brilliant scientists making new small molecules against previously undrugged targets with very interesting and complex biology. So I had a fabulous time at Millennium Takeda, but ultimately moved on to a company called H3 Biomedicine that was a Cambridge, Massachusetts-based small molecule precision oncology company utilizing data science and bioinformatics to pick targets and accelerate programs into the clinic. Again, enjoyed that experience, introduced a number of molecules into the clinic that, thankfully helped patients, and then ended up at Atlas Venture, which is a VC firm in the Cambridge, Mass area, as an entrepreneur in residence, and, worked with them on a few different ideas. Started a couple of companies, but one of them was Remix, and that's where I'm at today.
Thanks so much, Peter, for the wonderful intro, adding color there to your background. Before we dive too deep here as well, could you just provide a brief intro into Remix for us as well?
Yeah, absolutely. So, Remix Therapeutics is a small molecule platform company really built around three key areas of expertise. Data science and bioinformatics is one of those. The second is a set of unique multiplexed high-throughput screening assays. And the third is a set of proprietary chemical libraries that we've built, with the notion that we can tackle RNA processing to go after undruggable disease drivers. And, we're really excited with the progress that we've made and the early insights that we've made towards being able to do that effectively.
It's so exciting to see the progress that Remix has had so far. And taking things a step back, could you just tell us more about the genesis of Remix? What was the spark that started it all?
Yeah, it's interesting. I think it's after spending a period of my career in trying to build small molecules and even ADCs towards protein targets, and having good success there, but also realizing that there remain a number of targets that are hard to drug through directly interacting with proteins. And so knowing that there were a number of hard to drug targets out there that needed different types of approaches to go after them. So that was one angle. The second is, whilst at H3 Biomedicine, we worked very deeply in the area of RNA splicing, and not a new area, right? It's been known about for decades now how RNA is spliced and turned into mature mRNAs. But we worked on a project that was tackling a small molecule approach to interacting with the spliceosome, and just that deep experience over about 8 years of getting very familiar with that space, seeing the potential opportunity to work in that area of biology to find small molecule approaches. And so really the nexus of what I think the toolkit needed to look like to do this systematically was starting to come together. And, partnering with Kevin Bitterman at Atlas Venture, and Alex Harding, who was also at Atlas Venture at the time, coming up with this concept around a broad approach to build a chemistry effort towards drugging RNA processing to go after undruggable disease drivers.
Yeah, and I mean, while we're on the concept and topic, talking about the foundational story, I'd just be curious, Peter, from your perspective. A lot of folks talk about translation from academia. I think with Atlas, you saw both sides of that equation, right? Some of the builds coming out, some of the ones that you built in-house. Could you just even talk about the journey separating from Remix, just the journey of company formation through Atlas, your experience on that, maybe compared to some of the academic translation models that we've seen as previous podcast guests?
Yeah, I mean, I can speak to the experience that I had at Atlas, and others will have had different experiences, because it's a very versatile group in terms of how they go about building companies. I've seen them do asset-based companies or platform-based companies, and they all have quite different builds. What I like a lot about their approach is the ability to put initial seed investments into ideas and then pressure test through real work and lab-based work. Does this hypothesis have legs? And so trying to de-risk that investment. And so that's the approach that we took with Remix, and putting together a business plan that was really stage gated around achieving certain milestones. And importantly for my experience, that initial investment allowed me to hire a small group of scientists that had worked closely with me at a previous company. And that was, I think, instrumental in some of the early progress that we've made. Having a group of very talented scientists working very closely together allowed us to kick things off really quickly and make some rapid progress. And I think Atlas were happy with the progress that they saw, and we were eventually able to bring in additional investors to buy into the hypothesis and the company that we were building.
It's great to see the background, your experience as a founder going through that. I think that's fascinating. And just chatting more about Remix, more about your innovative approach here. I mean, Remix at its core is an RNA processing company utilizing a combination of computational biology, chemistry, and machine learning to identify RNA binding small molecules to co-op processing machinery and alter gene expression at its basic scores. But taking a step back, could you provide context for our audience and just share more about the drug development process in complex disease? I mean, there's so much from that perspective. I bet at Atlas, you got to see this problem thematically, but maybe could you share more with our audience just from your perspective, the challenges you see?
Yeah, I mean, it's really looking at the type of diseases that you want to impact, where there's a clear unmet medical need and the best approach to go after it. And of course, multiple modalities now in existence, not just small molecules, antibodies, RNAi, ASOs. And so it's really a case of looking at the spectrum of the disease you're looking to address and what might be the best approach and the best modality to go after it. And in the case of Remix, the expertise that we've built in-house is small molecule based. And so what are the right targets that would benefit from having orally available small molecules that can control how those targets are expressed? That's how we think about the diseases to go after, how we think about the targets that we want to impact. I'd say because we're in a newer area of small molecule modality, right? There's a lot of questions around how to interact with RNA processing, how to understand specificity and selectivity. So we're not in the business of trying to discover new disease targets. It's really, what are the targets that people have wanted to address because they're very closely linked or driving the disease, and how can you manipulate their expression? And so, we view that as de-risking on the target choice side, because we have high confidence that interacting with that target in a meaningful way will lead to disease modification. And so the way we want to go about doing that is through this RNA processing step. And that's really the puzzles that we needed to figure out to make progress.
I think that's been an amazing differentiating factor from your approach. And it just compared to the current limitations in protein targeting therapies, how your system is almost an upstream approach, I think has been really novel and great. I wanted to take a moment to dive into that specifically, give you a chance to just speak on this. I mean, so Peter, with the understanding of some of the more broader limitations in the space itself and the targets you're approaching, could you tell us a bit more about your approaches addressing some of these systemic challenges and just RNA targets? Maybe just share more about the benefits of working upstream of protein expression, like most small molecule companies that are targeting the space right now.
Yeah, and again, it's the lens that we take of which are the targets that have been hard to address through protein targeting approaches. So one class of proteins, and it's really not limited to this, but one class is transcription factors. And the example I can use is around our lead program that's targeting MIB. MIB is an oncogenic transcription factor. It's dysregulated in solid tumors and heme malignancies. Really a high value target that many people have tried to drug before, but it's been really challenging. There's no kind of binding site. It's a very disordered protein. There's no enzymatic activity that you could look to take advantage of to try and inhibit a reaction. And so it presents a real challenge to finding a small molecule that can ligand that protein target. Conversely, if we focus upstream at this RNA processing step, now we can find druggable sites in the RNA that can degrade the mRNA and then prevent the protein expression. So in some ways, the outcome's similar. You're preventing the protein from being expressed. The transcription factor is switched off, but getting there to do that is really quite different. And so we've shown promise in that target. We have additional ones where we're seeing some activities as well. And we think this is going to be a way to unlock a number of the disease drivers that haven't been addressed before.
I appreciate you bringing up the target discovery side. I think I would be really curious to hear from your perspective, Peter, as we're chatting high level overview here. Could you provide maybe a high level review of the platform itself? There must be a large library of potential, just protein folding and misfolding combinations. How as a company do you all turn that crank to enable this as a company, as a function, as an engine?
Yeah, it's a really cool process, and it leverages internal capabilities, but also external capabilities as well. So we really start with data, and primarily transcriptome data sets from diseased and normal samples, but also DNA sequencing data sets as well. We've accumulated a lot of that publicly available data, and so all of those consortia efforts to sequence cancer patients or other diseases to understand the genetic causes of disease, we've capitalized on the availability of that data. But importantly, we've also built our own databases as well. We now have terabytes of data that we've generated ourselves in-house through a number of different approaches. And so what you end up with is huge amounts of data that requires very powerful computational approaches to really sift through and pick out what we think are the appropriate target sites where we think small molecules could interact. So, we spent a long time building that database. We've made a real investment in computational biology and data sciences, I think leading the way in AI and machine learning approaches to understand variability in data sets to pick out these target sites. I think what was really exciting at the beginning of the company is the ability to see targets across multiple therapeutic areas. And so we saw a huge potential here for impacting a number of different diseases. And of course, one of the steps that you want to take is very quickly prioritizing the targets or the therapeutic areas that you want to work on so you can focus. And we've done that, but I think to really support the potential of this space, we decided to build multiplexed high throughput screening assays, I think unique in this area. So instead of, a traditional approach would be you pick your target, you run a high throughput screen against that target, and then you see if you can turn those hits into early hit to lead programs, understand selectivity. We built a screening assay that allows us to assess or screen hundreds, potentially thousands of targets at the same time in the same system. and importantly, doing this in a physiologically relevant setting. So using cells that read out function, but asking very specific questions about target sites that we'd computationally predicted. And out of that becomes another massive data set, over 100,000 compounds against hundreds of targets. What that allows you to do then is say, okay, this is a new area, where's the low-hanging fruit that you can act upon? And that's where our filtering step came in, of picking the targets that we wanted to work on, because there was a lot of opportunities for us. So I think that multiplex screening assay that we've built, and we've subsequently built additional screening assays, really has positioned us to capitalize on the last feature that we built, which is a proprietary chemical library. And we had to build that because we knew at the inception of the company that a lot of the high throughput screening decks that exist, very powerful chemistry toolkits that are available to the protein targeting world, we felt that they were not faithfully replicating or representing the kind of chemical space that this RNA processing arena needed to see. And some of the insights that we'd made, some of the knowledge that we had from prior lives at different companies, led to the realization that we needed to put our own chemical library together. And that's what we did. And it's kudos to really Atlas and the Column Group for seeing the need for building that and providing the finances to allow us to build that proprietary chemical library. So we think that's unique to us. A lot of those compounds we synthesized with the help of CRO partners for the first time, and it's beginning to bear fruit. And so an important investment that I think was really catapulted our drug discovery programs. And so, very simply, it's the data science and the computational approaches to pick out the targets, screening them against the chemical library, and then we have an understanding of advanced chemical matter, how it's acting, and how to optimize it towards taking it into the clinic.
Yeah, Peter, I'm really glad you brought up the holistic aspect of your platform, from discovery to development itself. And in particular, I think that's an incredibly novel approach. And I'm glad that Atlas and Column Group gave you the freedom to build that. And as there's a continuation question, I think, just out of curiosity, you as CEO, thinking about the structure of the team, the company itself, obviously you have computation, chemistry, along with the development and creation of these new therapies themselves. How do you think about building out this interdisciplinary team? What was your thought process while building or creating the early steps of Remix?
Yeah, we took the approach early on of just trying to find the best talented scientific minds who've got a hunger for making drugs, right, and real passion for developing new therapies for patients. We did spend, and we continue to spend, a lot of time thinking hard around what the culture we have today is, but importantly, how to evolve that culture moving forward. And so our goal was always and remains, is to find people who can add to the culture rather than just fit into the culture. And it makes us think deeply around personalities and how people can interact together, and how to bring in different personalities and different mindsets. So we're challenging each other from a place that's good, right? Wanting to make the science better, wanting to end up with making the medicines that we look to build. So that's, I talk really there more about kind of the cultural components that we thought about. Of course, in addition to that, is the expertise that we needed to bring in. And so that was that initial group of scientists that really provided that leadership, a head of data science, a head of the what we call biomolecular sciences and lead discovery group, a head of chemistry, and that was the expertise that we needed early on in the company to get things going. And as we've made progress, we've continued to bring in additional expertise and additional talent with that lens towards the culture and the evolution that the company needs.
Thanks so much for sharing that, Peter. I think with new models, it's always amazing to attack the growth, how you actually thought about the form and culture systemically. So really appreciate that. As just one follow-up continuation question, you discussed the terabytes of data that you pulled from yourself. And I mean, as you alluded to complex disease, it comes with a large variety of patient etiology. And I think I'd just be curious to know how does Remix plan to navigate this complexity in a way through data? Are you aiming to establish a panel of targets for all patients with a given disease, or perhaps a variety of molecularly defined patient segments for a given disease? I'd be curious about your approach going forward with the platform you're developing, some of the mindset that you and the team are thinking of.
Yeah, it's been a really important conversation that we've had internally and with our board and with our scientific advisors around target choices and diseases to go after. And our bias is towards molecularly defined diseases where you can understand ideally a genetic lesion that is causative to the disease state or associated with that disease state. That's going to allow a smaller company like Remix to be very disciplined around the assays that need to get built, but also the clinical trials that need to be executed in these smaller populations. Certainly, there are diseases without clear genetic linkages right now that there are good targets towards addressing them as well. We can see those kind of targets on our platform as well, and how we've tried to address those kind of targets is through the potential to do discovery or collaborations with other companies, with pharma companies that may have deep expertise in those areas of biology that Remix doesn't have. But we can see a path towards coming up with molecules that could address targets that are of interest to those groups.
Thanks so much, Peter. I just want to take a moment, pause halfway through our episode here, just to talk about the major challenges and accomplishments that Remix and you have faced building this amazing platform. Remix, as you all have been pioneering this exciting platform, and it's really in this simplistic form, it's representing an ambitious new therapeutic opportunity. And it's exciting to see you all approaching this, both from a team perspective, but also a modality and focused perspective. But looking back, Peter, what have been some of the greatest challenges you faced building it and going through Remix?
Yeah, it's been a really satisfying journey of trying to build the assays and the company, not without its challenges. Of course, there's a lot of enthusiasm right now for trying to find small molecules that can interact with RNA biology. A number of companies that are forging this path with us, right? All taking slightly different approaches or very different approaches. And I think as the community is continuing to progress ideas forward, learn directions that may not work out so well, and others, the sharing of information through meetings or other forums is just helping the industry to grow and start to solve some of these problems that we've all seen. For us in particular at Remix, with I'd say the screening assay that we looked to build, the multiplex nature of it, we don't think it had been done before. It's always possible it has been. We don't think it had been done before, certainly at the scale that we've been able to do it. And so that was a de novo build of a brand new screening technology at the scale that's really unprecedented. And that took time to figure out, right? We wanted to do our work in cells because we knew the machinery being present was important, and we wanted to read out functional changes because of that chemistry. One of the early iterations of that screen, we didn't see a lot of faithful recapitulation of our engineered setting versus the endogenous cell setting. So we were worried that we could run high throughput screens in this multiplexed assay system, and we could find some cool chemistry, but then trying to translate that into the right disease models might fail if there was if the recapitulation wasn't there. And in probably the first iteration, it looked okay, but really not as to the level that we wanted to. So we had to optimize that. And it took a lot of work by a really talented group of scientists at Remix to play around with, different constructs, different ways to measure the activities, such that now we see very high fidelity between our engineered assay systems and that translating into relevant disease models. We could have decided to stop that and take the easier approach of doing single target screens, but we just thought this was going to be such a powerful technology that doing it and getting it right was going to be really important to the beginning of the company. And I'm glad that we, and in particular the scientists in the lab, really stuck with it and problem solved to get around some of those initial challenges that we saw.
Can certainly perceive how challenging that must have been. And as you said, Peter, this was a rewarding or just a satisfying build as itself and on its own. And so I think you alluded to some of the successes that you all have had thus far, building the platform and program. But any other milestones or key achievements that you'd want to highlight just through the journey looking back that were special to you or important to point out?
Yeah, and there's probably too many to list out. I think another is we want to ensure that the chemistry that we're working with is acting in the way it's intended, and we can get a lot of that information from our screening technology, but we also want to understand the biophysics and the structural biology and how these molecules are really sitting at their targets of interest. So we've built multi-protein RNA complexes now in a cell-free setting, and really being able to recapitulate the activity that we see in cells, but now in a cell-free setting. It allows us to understand, compound on rates, compound off rates, binding affinities. And, it's just the first time we saw some of this data, it was remarkable to see, right? Exactly what we were seeing in the functional assays, understanding how that's happening at a molecular level. It's given us a ton of insights into how to do this repeatedly for additional targets. So I do remember the day when we started to see that data come off, and just the real excitement in the team of seeing everything that we'd been kind of working towards in terms of understanding the chemistry and how it works, playing out in a very orthogonal assay system and giving us the same kind of information. So we have other days where we've seen some really cool stuff, but that's one of them in particular.
That's incredibly exciting. I'm glad you and the team got to share that awesome moment. I'm so excited that we got to chat about your approach to date, the background, the history, the growth of Remix. But I want to take things to looking forward here, Peter. And just to start simply, what's coming next for Remix? What are the next few steps here? How can our audience kind of follow along or look forward to what y'all are building in the future?
Yeah, moving molecules into the clinic, another one of those examples where we had the wow, that's amazing moment is the first tumor xenograft data that we got back for our lead MIB oncology program, and seeing tumor aggressions at well-tolerated doses in translationally relevant models was just amazing for us all to see, extremely exciting. So what's next for us is moving that program into clinical development. We have additional programs coming behind it. And so I think really excited to see how these molecules perform in humans. We've done all the right studies to do those experiments, and that will be really the next important data sets that we generate at the company.
Looking forward to seeing your progression as it goes on. Thinking more broadly towards the future and just the applications of Remix, are there any other additional enabling technologies you're excited for with such an interdisciplinary team, what you had to build to make Remix what it is today? Are there any current or upcoming innovations, say in SynBio or next gen sequencing, comp bio, that you just be excited that could accelerate the current work that Remix is doing?
Yeah, absolutely. I mean, I think the application of computational approaches and machine learning across the drug discovery continuum is something that is making a big impact in the work that we do, from the target discovery, from the target evaluation through to how the chemistry is working and applying it to chemical matter, and then into the clinic. And so I think the investments that we continue to make there and leverage the latest and greatest on the computational side is going to continue to accelerate what we can do and what the industry can do.
Fantastic, Peter. Well, just a few rapid fire questions before we close up here. Just imagine it's 2050, right? We're looking into the future with so much happening in the mRNA space right now. And like you said, so many approaches. I'd be curious from your perspective, could you describe the mRNA field by 2050? What challenges have we addressed? What diseases have we treated? Where are we going from now?
Yeah, I mean, it's a long way in the future, but I can see areas that the industry is starting to solve problems that will have really become real at that point. mRNA gained a ton of attention through the COVID pandemic. The ability to appreciate what that technology has done and now apply mRNA as a therapeutic itself to different diseases, I think we'll have overcome many of the delivery challenges, the sustainability of expression. I think applying technologies in the small molecule space to induce mRNA degradation, as we are doing at Remix, we see a path towards boosting mRNA expression as well. I think that will be a really powerful technology for a number of diseases that don't have a useful approach yet. But then even beyond mRNAs, there are groups looking at different types of RNA, tRNAs, long non-coding RNAs, that will all find their place in the treatment paradigms for different disease states. And so, what's that, 20, 30 years time, there'll be more modalities, more drugs on the market, and more ways that we can help patients and address these diseases that haven't really effectively been addressed yet.
So great to see from your perspective, Peter. Would love to flip it to you for a minute. In 2050, where do you see Remix?
We are on our path, right? We're moving into the clinic shortly. Hopefully this plays out just as we see a repeatable machine to come up with new small molecule approaches to degrade mRNA, new small molecule approaches to boost RNA expression. We think the platform has the ability to touch a number of different therapeutic areas. So, our goal and our vision is to make these medicines to help patients. And I think it's just continuing to build our sustainable pipeline to make sure that we're doing that in a very efficient way. And that, however long that takes, as long as we can keep ourselves around here, we'll be very excited to have achieved that.
Of course, and I'm incredibly excited to see the journey and how you all fit into the space broadly going forward. So thanks again, Peter, for your time. Any closing thoughts here, shameless plugs that you'd like to end our episode with here?
No, I've really appreciated the conversation. I think what we're doing at Remix is really cutting edge. There's a cadre of companies here that are trying to do small molecules and RNA. It's really exciting to be part of that group of companies that are pushing things forward. I think we've made a number of really important insights that are allowing us to do this very systematically, think deeply about target choices, and pick the right chemistries to go after to develop medicines. And I think we're doing this with a team of scientists and co-workers that are really very motivated to do cutting edge science and doing it in the right way. So we talk about, finding good people who are doing great science and just building that team towards a common shared goal. And I'm really proud of the culture that we've been able to build so far here at Remix. People are just trying to do the right thing for science and turn that into medicine. I'm really looking forward to how new folk that come into the company can help us continue to evolve, because I never want us to stand still, and I want us to continue to challenge each other, come up with the right kind of questions and try and answer the right questions. And ultimately with that goal, making medicines.
I'm so excited to see that you and the team share the sentiment of a patient-centric company and just all the focus towards that translation and that impact. So love to hear that, Peter. How can our audience just follow you along, hear more about your work, and just join the Remix journey?
Yeah, we've started to share more of our science at conferences. So we expect to be at more conferences, RNA-centric conferences in the coming year. We're on LinkedIn, we're on Twitter as well. And so I think the number of different ways that we can be found, and we try to keep the public abreast of kind of the progress that we're making.
Thanks again so much for your time. Really grateful for this episode. So thank you again.
Great. Thanks, Drew. Really appreciate it.